Kingdom Monera, right? So let's talk about Kingdom Monera. Coming to this Kingdom Monera consists of unicellular, prokaryotic, organisms. So coming to the types of Kingdom Monera, we have two things. One is archaebacteria and other one is eubacteria. First thing regarding eubacteria. Eubacteria we otherwise call it as true bacteria. They are characterized by the presence of rigid cell wall. So the main difference between archaea and eubacteria is cell wall. So eubacteria have peptidoglycan. So as cell wall, in the cell wall material. So eubacteria will include the things like bacteria, cyanobacteria, which is blue-green algae. We otherwise call it as blue-green algae. Next, coming to the bacteria. So bacteria, so you have a statement here. All bacterians are monerans, but not all monerans are bacteria, right? So these are the sole members of Kingdom Monera. It was Anton von Leeuwenhoek who first observed bacteria. So he is the father of microbiology. So from pond water and tartar of teeth, he saw using the microscope made by him because he himself is the lens maker. The term bacteria was proposed by Ehrenberg in 1829. So coming to the next thing, distribution. So coming to the part of distribution, they are ubiquitous or cosmopolitan. So they found in air, soil and water. The reasons why they are found in all the habitats is because of small size, rapid division, rapid reproduction, numerous modes of nutrition, and we have the airborne endospores. So which during unfavorable conditions. So bacterial cell has been seen in seed beds in atmosphere. They also live in extreme habitats like hot springs, deserts, snow, right? And so many are live as parasites also. So coming to the cell structure, they have a rigid structure. Which is made up of peptidoglycan. And they are motile because the flagella is present, which is made up of flagellin. and they have 70s ribosomes. They have cytoplasm, DNA, presence of free DNA, membrane bounded cell organelles are absent. So, coming to the classification based on morphology. Classification based on morphology. So, we have cocci. Cocci is plural. The singular is coccus. They are oval or spherical but without flagella. So, they don't have flagella actually. The spherical will be the diameter of 1 micrometer to 5 micrometers. They may occur in single. So, if they occur single, we call it as monococcus. If they occur in pairs, we can call it as diplococcus. If they occur as 4, we can call it as tetracoccus. If they occur as cluster, staphylococcus. If they occur as chain, streptococcus. Right? And if they are arranged cells in the arrangement of cube, then we can call it as Sarcina. And next coming to rod shaped bacilli, and singular we call it as bacillus. So they are the rod shaped bacteria. The length will be 1.5 micrometers to, and width will be 0.5 micrometers. Usually you will be having single rod shape. Monobacillus. So if it occurs in pairs, diplobacillus. If they occur in chain, streptobacillus. If they occur as layers, palisade bacillus. And next, spirillum. And plural is spirilla. So twisted like screw, and they are free single cells. Spiral shaped, and they are the large side reaching up to 20 micrometers also. So here we have so these are the flagella, spirochetae. And we have the next thing, vibrio, comma shaped. So example, Vibrio cholera. And mycelial or filamentous. So they are like extensive chains of rods. So actinomyces. So this will produce antibiotics. Next, coming to the nutrition. So the nutrition of bacteria is very simple, and they are very complex in behavior compared to many other organisms. Bacteria as a group show most extensive metabolic diversity. Most extensive metabolic diversity. So bacteria shows autotroph and heterotrophs. First thing, autotrophs. Next thing, heterotrophs. So coming to the autotroph, we have photoautotroph. So which captures the sunlight, which contains bacteriochlorophyll. So example, purple non-sulfur bacteria, green sulfur bacteria, so bacteria photosynthesis is anoxygenic. So usually this type of photosynthesis, the source will be H2S, not H2O. So purple non-sulfur and green sulfur bacteria, these are the photosynthetic. And next, chemosynthetic. Coming to the chemosynthetic, so bacteria oxidize numerous type of inorganic compounds and trap the energy in molecules like ATP. For example, we have the nitrifying bacteria. Such as Nitrococcus, Nitrosomonas, Nitrobacter. We have the iron bacteria. Sulfur oxidizing bacteria. And heterotrophs. Coming to the heterotrophs, so the bacteria is ready-made, they take the ready-made organic food from different sources. Based on the nature of the source, heterotrophs are divided again into three categories. They are parasites, saprophytes, and symbionts. Coming to the parasitic bacteria, so they attack the living plants and animals, obtain organic nutrients from them. These bacteria causes diseases such as pathogens, like we have Salmonella typhimurium, which will infect the humans and will produce, and it gets the organic food from the host and causes the typhoid fever. Salmonella typhimurium. Saprophytic, these are the decomposing bacteria. They act on animal excreta, dead plants and animals. Symbiotes, for example, Rhizobium. Frankia. These are present in the root nodules And the bacteria can able to fix the atmospheric nitrogen also in leguminous plant as an organic food for the bacteria. The mutually beneficial relationship between two types of organisms, we call it as symbiotic association. Next thing is respiration in bacteria. So the respiration in bacteria will be, there are two types: aerobic, oxygen dependent; anaerobic. So you have aerobic, anaerobic. Coming to the aerobic, so respiration is energy creating process where organic food is broken down and energy is released. So, and energy is stored in ATP, and these high energy molecules are synthesized various biomolecules required by the cell. Bacteria with aerobic, we call it as aerobes; anaerobic process, we call it as anaerobes. Next, reproduction. Coming to the reproduction, they will produce sexual as well as asexual, right? So coming to the sexual, we also call it as asexual. Sexual, we also call it as parasexual. Asexual occurs as in two ways: binary fission and endospore formation. Bacteria will mainly produce by binary fission. During unfavorable condition, they produce spores. They reproduce by the sort of sexual reproduction by adapting primitive type of DNA transfer from one bacteria to the other. Okay, so the sexual reproduction, this process we call it as conjugation. So transfer of genetic material with the help of sex pillai. And next, coming to cyanobacteria. So coming to the cyanobacteria, the kingdom Monera has a separate division called cyanophyta, earlier called blue-green algae. They have very similar to bacteria, but the term cyanobacteria, they also differ from bacteria in some characters. Some common examples of cyanobacteria are Anabina, nostoc, Rivularia, Spirulina. So they have adapted themselves to grow in wide variety of habitats, fresh and seawater. The polluted water they grow, a huge population causes water blooms. They also grow in moist soils. Single-celled, we call it as Crococcus, and colonies we call it as Microcystis and Gliocepsa. Filamentous will be seen in Anabina, nostoc, oscillatoria. So you can able to see them as in the NCRT they have given a picture like this. In the heterocyst, the nitrogen fixation will take place. So the entire things are actually covered as a mucilaginous sheet. So this is the heterocyst seen in nostoc. And they are prokaryotic. The colonies are generally surrounded by the flagellal sheet, and flagella is actually gelatinous sheet. The flagella is actually absent for this. They often form blooms in polluted water bodies. Some of the organisms can able to fix the atmospheric nitrogen also. So we call that parts as heterocyst. So these are the heterocyst. So Anabina, nostoc, oscillatoria, rivularia. And also the specialized cells called akinetes. Primary helps, they have specialized cells called akinetes that helps in storage, food storage, and all of them are photosynthetic. Different types of light capturing pigments will be there, and it has mainly chlorophyll A because the chief photosynthetic pigment. Without this there is no photosynthesis. And blue pigment called phycocyanin. Phycocyanin and C phycoerythrin. Yellow pigments called xanthophylls. So chlorophyll A, phycocyanin, phycoerythrin, xanthophylls, these are the pigments that is present. And vegetatively they reproduce by fragmentation. Simple body without tissue. Akinetes, heterocyst are also involved in vegetative reproduction. Sexual reproduction is actually absent in cyanobacteria. So let's talk about the other thing called archaebacteria. So coming to this thing of archaebacteria, so they are the members of this domain R, we call it as archae. They are the most ancient and most primitive prokaryotes. They are found in high stress environments like high salt content, high temperature. So hot springs, ocean thermal vents. So the cellular plasma membrane has specialized lipids and cell wall made up of proteins and the non-cellulosic polysaccharides which can withstand high stress. They have special lipids and complex polysaccharides which is non-cellulosic. So that's how they can able to sustain more pressure. Most of them are chemoautotrophs. There is no parasite. Since that environment do not have host, so no parasites. They don't act as any parasites. Methanogens. We have the categories. Methanogens. They live in anaerobic environment. The anaerobes like swamp, deep sewage, gut of the ruminant like cows and buffaloes. They produce 65% of atmospheric methane. They are responsible for production of methane, biogas, from the dung of animals. So, example, Methanococcus. And halophiles. Coming to the halophiles, they live in salty habitats. And mostly these are also anaerobes. They have chemical called halorhodopsin to pump the chlorides into the cell and prevent cellular dehydration. They also have the chemicals to synthesize ATP in the presence of light. So, example, halobacteria. And thermoacidophiles. So coming to thermoacidophiles, they can survive in high temperature habitat like 65 to 80 degrees Celsius, like sulfur springs, volcanoes, submarine thermal vents, and natural geysers. They also survive in a high pH. We have thermoproteus and thermobacillus. And the next category is mycoplasma. We otherwise call it as PPLO, pleuropneumonia like organisms. These are the monerans, belong to the class molecules. They are discovered by knockout and rocks. So in the lung fluid of the cattle affected by bovine pleuropneumonia, these cells don't have no cell wall, and they are of 0.1 to 0.15 micrometers in size. The plasma membrane is the outermost boundary. The cell change its shape and often. So like the prokaryotes, they have no any nucleus and membrane bounded organelles like mitochondria, plastid, vacuoles, Golgi apparatus, cytoskeleton. So ribosomes are 70s. So they have double-stranded circular DNA. Cells do not have flagella. Enzymes have bounded to plasma membrane, and some enzymes occur in cytosol also. They lead a parasitic existence in animals and plants. Some of the plant diseases caused by mycoplasma are potato purple dwarf, mulberry plants like witch's brooms in legume plants, witch's broom in legume plants, and in papaya bunchy top disease, and pleuropneumonia in human and cattle. Coming to the economic importance of bacteria of kingdom Monera. Coming to the economic importance, heterotrophic bacteria are the most abundant. The majority are the important decomposers. Bacteria of rhizobium, like they are the important decomposers. Rhizobium that mainly help for fixing atmospheric nitrogen. So fixing nitrogen in the root nodules. Nostoc, Anabina, they are also used as a biofertilizers. Lactobacillus, so used for lab bacteria, used for production of lactic acid. So bacterium named Propionibacterium shermani, used for the production of Swiss cheese. Use it to produce Swiss cheese. Actinomycetes, use it to produce antibiotics. Acetobacter aceti, acetic acid. Clostridium vitilicum, Butyric acid. Methanobacterium, biogas. Bacillus thuringiensis, BT, biopesticide. Thermus aquaticus. Biotechnology. Agrobacterium. Plant biotechnology. So these are the economical importance.